All right, so let's try this out. Okay, so today we are talking, that's the wrong class. Today we are talking about proteins. So you guys did nucleic acids yesterday. How'd that go? Good? Okay. So, what is this? All right. So today it is all about proteins. Now, I only have one page, one concept map, and we're doing the announcements. Okay. I think that's unpaused from where we were. Morning announcements, all that fun stuff. All right. So, proteins. There's my marker. Is this going to let me move this over? I don't want to mess with this right now. All right. So, we're going to start out talking specifically about protein functions because protein functions are literally almost everything that we do kind of boils down. There we go. That's not what I wanted. There we go. Boils down to proteins. So, the first function we're going to talk about is enzymes. Or I guess enzymes are an example of something that reduces activation energy. So, if this little doohickey right here is our enzyme, this is our protein. This is a reactant that is going into a chemical reaction. That guy comes in here. Then we've got our enzyme substrate complex. And after our enzyme substrate complex, we've got our reactant or our products. Essentially, enzymes are proteins that reduce activation energy by giving the chemical reaction a place to take place in. That place is called the active site. Also, little side note, all proteins, or not all proteins, all enzymes usually end in that suffix ase. So if you're taking the AP exam and you see a really, really big long word and it's kind of freaks you out because you don't know what it is or you don't recognize it on site, but it ends in ase, that essentially means it's an enzyme. And most enzymes are named after the reaction that takes place within them. So if you see ATP synthase, it's an enzyme that synthesizes ATP. That's function number one. I'm just going to make this real long. Go back up. All right. Next protein function, defense. The example I have here are antibodies. So antibodies are proteins that exist on the outside of our cells that help us identify what is us and what is foreign, what we should attack. We've also got our proteins as storage. And the example I have here is ovalbumin, which is essentially in eggs, like all eggs, not necessarily just chicken eggs, all eggs, period. Like hard, like crackable eggs. There is like a store of proteins in there called ovalbumin. I believe it's the white stuff. It might be the yolks. But essentially, all the proteins, all the nutrients, all the stuff that the growing organism would need is stored in that ovalbumin. We've got proteins as transport. So we've got like cell membrane proteins. Cell membrane proteins. So all of the proteins that are kind of embedded in a cell membrane that help stuff get in and out. We also have stuff like hemoglobin. Hemoglobin essentially is the protein in your red blood cells that carries oxygen from one place to another. There are also hormonal proteins. So, like human sex hormones, steroids, that kind of stuff, those are lipids. But stuff like insulin is a protein, and that's the protein that helps regulate blood sugar. That's a really, really important protein. We've got receptors. So, like neurotransmitters, things that leave the end of one neuron and send the signal to the next neuron, those are those are like neurotransmitters, and what receives those neurotransmitters on the end of cells are receptor proteins. So, their job is mainly just to receive stimuli. That. Proteins are also really important for movement. There are motor proteins. So proteins whose main job is to move. So like I know there's one specific protein that sits in a cell membrane and kind of spins, and that spinning allows ATP to be produced, I think. So motor proteins, also actin and myosin. Actin and myosin are the proteins in your muscles that allow muscle contraction to happen. So all your muscle movements are due to actin and myosin. So those are some pretty important proteins. And then one last one is structure. So let's see, keratin. That's I misspelled it twice. Keratin is the protein or that strengthens your hair, skin, and nails. A lot of people take keratin as a supplement to help try to improve that kind of stuff. I know keratin is in a lot of shampoos that are dedicated to like strengthening like already straight hair. So keratin is a structural protein. Any questions about the different functions that proteins carry out? All right. So we're going to go way back up here. So proteins make up about 50% of a cell's dry mass, which means if we take a cell and we dry it out and we weigh it, about 50% of that weight is proteins. Scientists usually do that by taking a protein or taking a cell, spinning it down, weighing that, and injecting a bunch of stuff that'll dissolve everything but proteins, and then spinning it down and weighing that, and they can from that can determine that about 50% of a cell's dry mass is like just proteins. We talked a little bit already about enzymes. Essentially, I'm going to add the word catalyst here. So enzymes are our biological catalysts. I've already told you that they are the ACEs. Catalysts are essentially anything that speeds up a reaction by reducing the amount of activation energy it's needed. Enzymes are just catalysts that exist within a biological space. So, from there, actually, yeah. So, we talked about monomers and polymers. So, proteins are polymers made up of amino acids. I have two things to come off of this. So, looking specifically at amino acids, they are essentially organic molecules with an amine group a carboxyl group and an R group. That R group is essentially just one of 20 different like possible chemical side chains that exist on amino acids. So we're going to take that. We're just going to put it way down here. So the way that looks so this is what our amino acid looks like. We have our amine group over here with the N's and the two H's, and we have the carboxyl group with the double bonded oxygen and the OH group. And then we have the R group, which is what gives each individual amino acid its identity. This central carbon right here is called the alpha carbon. Or there we go, alpha carbon. They essentially just named it that because it's the central carbon on all of the like every amino acid has that one central carbon structurally. It's the one thing that the amine group and the carboxyl group and the R group all branch off of. So the R group is what makes each amino acid different. And there's 20 of them total. So if there are 20 different R groups, that means there are 20 different amino acids. So all of our thousands and thousands and thousands and thousands of proteins all boil down to 20 amino acids. If you take a biochemistry class in college, they might have you memorize them. I did not have to memorize them. So these 20 groups, or these 20 amino acids can fit into 3 groups. Yes. Yep, different orders and different shapes, and we'll get into those different kinds of shapings at the end of this. So yeah, no, it's literally just how many and what order of amino acids that makes each protein different. Yep, we can have the same number of amino acids in a very similar order, making two very different proteins. Just like you can have like the same chemical composition of a glucose polymer and cellulose and starch being completely different, even though they're made of the exact same composition. It all boils down to which shapes form because which bonds form. So, our three different types of amino acids, we've got nonpolar hydrophobic, we've got polar hydrophilic, and we have charged hydrophilic. Just because these molecules are hydrophilic does not mean that they will dissolve in water. These guys are too big to dissolve in water, unless you try really, really hard or you melt them, but we're not going to mess with melting them. So, nonpolar hydrophobic, polar hydrophilic, and charged hydrophilic. These amino acids are connected by peptide bonds. Now, so every amino acid in every protein is connected to another amino acid by a peptide bond. Now, peptide bonds are just covalent bonds formed through dehydration reactions. Between amino acids. So the only time something will be called a peptide bond is if it is bonding two amino acids together. Because scientists like to name some things a million times. They couldn't just call it a covalent bond. When it comes to amino acids, they have a special name for it. Reactions, sorry. Reactions. And if we remember from our first like carbon compounds lecture, we've got our two amino acids right here. Got our these guys come together to form one chain. We've got our N, our C, our C, N, our C, our C, our O. This has two hydrogens coming off of it. This has our R group. This guy has the double bonded oxygen. R group, double bonded oxygen, OH. Yeah. And when we do this reaction, we get water. So this is the same kind of reaction that we talked about with carbohydrates, where we're losing water to link two together, but we have our amino acids with our nitrogens instead of carbons. That looks kind of messy, but I usually divide, or I identify our amino acids by our R group. So I'm like, oh, there's an amino acid there. There's a nitrogen. So that's an amino acid. That's an amino acid. So if you cut them off at the nitrogens and then make sure that each one only has one R group, that's usually how you can detect how many amino acids are in a certain chain, especially if it looks like chemical alphabet soup. So a chain of amino acids is called a polypeptide. The difference between a polypeptide and a protein is that a protein is a functional polypeptide with a 3D shape. So if you think about like a wedding cake, like it's all decorated, it's all stacked up. A polypeptide, if that cake was a protein, a polypeptide would be like the cake batter. All of the ingredients are there. It's just not functional as a cake, and it's not put together yet. It doesn't have a shape. So sometimes you'll get polypeptide and protein kind of interchanged, but a polypeptide doesn't have the shape or the function yet. It's just the chain of amino acids. So many peptides, peptide and amino acid are sometimes used interchangeably as well. That's why they're called peptide bonds, because they're bonds between peptides, but amino acids have multiple names. So everybody get the difference between a protein and a polypeptide? Cool. So now we get to get into the fun stuff, which is protein shape. Now, protein shape determines its function. Without a shape, a protein has no function, period, end of story. This is kind of what a lot of biochemistry boils down to. This is a protein. It's not, if it's shaped wrong, then we're done. It's garbage. A lot of diseases and disorders come from misshapen proteins. Lactose intolerant, if the lactase enzyme is shaped differently or off a little bit, then that protein can't digest lactase, and someone is lactose intolerant because lactase doesn't work to digest lactose, so you can't use it. Sickle cell anemia is if I think it's you switch one amino acid on the hemoglobin protein. That one amino acid changes the structure so that hemoglobin molecule can't carry oxygen as well as it could have if it was completely normal. So, protein shape determining function. Protein shape determines function. I'm writing it twice because I want you to write it twice in that key terms page at the front of your notebooks. And if you want to go ahead and write that ACE equals enzyme 2, I won't be mad at you because that one's a pretty important key term too. So protein shape determines function. Without a proper shape, the protein can't perform its given function. Protein shape determines function. There are 4 levels to protein structure. And we are going to talk about those 4 levels of protein structure, starting with our primary level. So our primary structure so the first thing that kind of helps contribute to protein shape is just the linear chain of amino acids. So the number and order of amino acids is our first level of defense for our protein shape. That order is determined by genetics. So all proteins are coded for in DNA. So the DNA gives the directions about which order those amino acids are going to be formed in. So our primary structure is our linear chain of amino acids, and the order is determined by genetic information. And that's level 1. Level 2, or secondary structure, is regions stabilized by hydrogen bonds between atoms of the backbone. So the backbone of a polypeptide is essentially what I drew when I drew the dehydration reaction. So it's everything but the R group. So it's that NCC, NCC, NCC all the way through. And those nitrogens and carbons can form hydrogen bonds. And those hydrogen bonds can lead to alpha helices or helixes, I guess. And beta pleated sheets. So in an alpha helix, essentially that big long string of amino acids can twist up like this. And in our beta pleated sheets, they essentially do that. I can show you a more 3D rendered picture that's not just me drawing on the board after we get through all of our levels. So that's primary and secondary. We also have our third level. Our third level or our tertiary structure. I did a brief overview of this with my Bio 1 students, and the word tertiary blew their minds. Like they didn't know that there was something that comes after secondary, and the fact that it was tertiary, like some of them just couldn't grasp it. But anyway, we have our tertiary structure, which is our 3D shape, which is stabilized by side chain interactions. So our primary structure is determined by genetic information. Our secondary structure is provided by the carbon backbone. But the third level of structure is where those R groups come in handy. It's where the fact that there's the hydrophilic and hydrophobic R groups. Those R groups is what determines our 3D shape. So, let's see, hydrophobic. That's a weird word to write in cursive. Hydrophobic interactions exclude nonpolar molecules. So if you've got let's see. So if you've got like a group of those hydrophobic R groups in a row, those will kind of like fold in on one another so they don't really touch the outside, so they don't have to mess with water. It's kind of also that like hydrophobic interactions are kind of how phospholipids work, like forming cell membranes. So those hydrophobic interactions will want to like close in on one another. The next one is actually really cool. They are disulfide bridges. So essentially in a disulfide bridge, covalent bonds form between sulfhydryl group. So some of those amino acid side chains have sulfhydryl groups in them. If you remember what a sulfhydryl group, I believe it's just a sulfur and 2 hydrogens. So if this is our structure, it's done its secondary structure. We're working on our third. And there's a sulfhydryl group here, and there's a sulfhydryl group here. A disulfide bridge will form, and then it'll kind of look like, so we've got that covalent bond right there. It'll kind of do that. Or somehow in 3D space bend to make that happen. Any questions about tertiary structure? So some proteins are done after this. Some, like when it comes to some proteins, they're like, okay, you've got your 3D shape, go exist. But sometimes they have a fourth level. So quaternary structure is optional. Not all proteins need it. But essentially, it's when 2 or more polypeptides come together. An example of this is hemoglobin. So hemoglobin has 2 subparts, alpha hemoglobin and beta hemoglobin. And so each portion, alpha beta globin, heme beta globin, is its own protein, and those proteins come together to form one giant hemoglobin molecule. So let's see here. We've got I learned I could do this the other day. It's kind of blurry, but you can get the gist of it. So if this is our primary structure, it's our order of amino acids. So There's an amino acid right there. There's an amino acid right there. Each one has a nitrogen and an R group. Each of those R groups is different. Our secondary level of structure, we've got our carbon backbones forming hydrogen bonds with one another. So like here, 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 and probably there. Those hydrogen bonds are what kind of keeps it in it. That's in that little coil. Our tertiary structure. So if this right here is this right there, we've got a bunch of those and maybe some sheets. That's one of those zigzaggy sheet things. And it all kind of like, honestly kind of looks like you took a pipe cleaner and you crumbled it up in your hands. And then our quaternary structure, we got one here, one here, one here, and one here. And all of those proteins come together to form one functioning protein. Yes. Yep. So not all of the protein will be in a helix or sheet. I think there are a couple other ones that aren't as common, but most of the time it'll take a bunch of these, package them together, and then when that package is somewhat stable, it'll bend and twist into what it needs to be. And odds are it'll happen kind of simultaneously, like as it's being built, but it's easier to think of it in steps. Good question. Any other questions? All right. There is an exit ticket on Canvas, just like there was for our last concept map. So, yeah, no, get that done. And then on Monday, or I guess you guys would be here on Tuesday. Let's see. So you guys are doing the progress questions on Monday. The link will be on it'll be through AP Canvas, and then we will take all of this information that we've learned the past couple days and kind of synthesize it into one big common like sheet on Tuesday when we're in class. Cool. All right. And then we get to start talking about cells. Super fun.